Hi, I'm Krissa Watson, and this is my mini exam video 4, and I'll be discussing prompt 2, which is the process of transcription. Transcription is the process of gene expression where genetic information in the form of DNA is converted to RNA. Prokaryotes are polycistronic, which means that they have one RNA coding region that can produce multiple proteins. The first part of transcription is initiation. To begin this process, RNA polymerase will bind to the promoter region of a strand of DNA. RNA polymerase is able to bind to the promoter region with the help of sigma factors. When adding a sigma factor to an RNA polymerase, it will produce a halo enzyme. The sigma factors are able to identify two important regions of the promoter region, the -35 region and the -10 box. Once RNA polymerase is bonded to these regions, it will move upstream from the 3' to the 5' direction and begin transcription at the +1 position. The next step of transcription is elongation. During elongation, the coding strand and the template strand become separated. The template strand is read from the 3' to 5' direction, while RNTPs are added in the 5' to 3' direction, because the base pairs are complementary. These base pairs form together through dehydration synthesis and form phosphodiester bonds. Halo enzymes are also converted to core enzymes during elongation through the loss of the sigma factor. The final step of transcription is termination. In this process, there is something that signals the end of RNA transcription. In prokaryotes, a common signal for the end of transcription is termination hairpins, which occur when there is an inverted repeat at the end of the RNA sequence. The inverted repeats allow the base pairs within the strand to bond together and stops RNA polymerase. At the end of the template DNA strand, you can find a poly-A tail, which is a long chain of adenines that form a bond with uracils, which is a very weak bond that allows the DNA and the RNA to separate. So what would happen with the deletion of a -10 box in a gene that codes for an enzyme that metabolizes sugar? In this case, RNA polymerase with the sigma factor would not be able to bond to the promoter region because it's missing the -10 box. Since the RNA polymerase would not be able to bond to the DNA, it could not transcribe the template strand. Therefore, an enzyme would not be produced, meaning the individual would likely be sugar intolerant.